Dr. Prasanji,
Good evening, everyone. I am Prasanjit Roy from Agartala Government Medical College.
Dr. Soni,
from second year, CMC College.
Dr. Harshad.
Hi, I'm Dr. Harshad, second year, KD Hub Hospital Matter.
Okay, Prithvi, I don't think is there.
Not there.
All right.
So,
welcome all of you.
and Shiva Ranjini will be moderating the sessions. Our webinar hosts are Dr. Anita Borges ma'am and Dr. Sumit Bishal sir. These are few reminders. If the connection gets interrupted or you have low or no sound, video freezing or video lag, kindly leave the meeting and rejoin with the same link. If the problem persists, please check your internet connection. The poll questions should be answered in the chat box.
Did not, but still these are the questions that were asked. Okay, and I think Ima and I think we have some answers in the chat box.
Yes.
Okay, so the first question was, since the RB and TP53 tumor suppressor genes are so important in the cell cycle, why do RB and TP53 null mice develop normally? Now, before we get to the answers in the chat box, I just want to ask, Chitra, you're a third year.
Yes, ma'am.
So can you tell everybody what is the meaning of a null mouse? RB null or TP53 null? Do you know what is the meaning of a null mouse?
The mutation is not present. The
Mutation is not present. What does that mean?
The RB gene, that is normally there, it has undergone it is a tumor suppressor gene, but that tumor suppressor gene is not working.
Is not working. Okay. But what do you mean by RB null? What is the meaning of null?
Not present.
Not present. How many retinoblastoma genes are there in every cell?
Two.
So what do you mean by null?
Both.
Both. So it means that both the genes are not working, right?
Yes.
Yes. So not working means that they're not there, because these mice are not born with no retinoblastoma genes or no T. They are created in the lab. Okay. So these are known as knockout mice, and I'm sure that you will, if any of you bother to read this, all the different paragraphs in your chapter. I don't know. Somebody told me last time that it was chapter 7. Is it chapter 7 in Robbins neoplasia, or is it chapter 6? Yeah.
7, I think, ma'am. 7,
you think? Okay. The older Robbins had it in chapter 6, but whatever. So if you read that, everything that I have discussed is with respect to a standard textbook of pathology, which is Robbins. And what I tried to do the last time is to take what Robbins is talking about and dissect it so that we will understand what is written over there. Now, Robbins tells you that the retinoblastoma gene and the TP53 gene, both of them are very important in the cell cycle. So much so that if you lose, the Knutson's hypothesis? Yes. If you lose both retinoblastoma genes, then you start to produce tumors. Yes. And those tumors are retinoblastomas. If you lose the TP53 gene is mutated in both copies of that are present. Yes, it's a tumor suppressor gene. You should get very disordered growth, because TP53 is involved in the cell cycle in which it promotes repair, that in the sense that it stops the cell cycle in G1 if it finds that there are abnormal mutations or there is something in the DNA that is abnormal. It stops it and allows the repair machinery to repair this DNA. If repair is not possible, then TP53 will push that cell into apoptosis, and that's why it is called the guardian of the genome. It does not want cells that have mutations or other defects in the DNA during replication to replicate so that it's passed on into the daughter cells. Now, if this is such an important function, which it is in your cell cycle, both retinoblastoma and P53, how is it that mice which are made in the lab, in which they have knocked out the RB gene and the P53 gene, or it's usually one knockout for RB and another knockout mouse for P53. If it's so important, how come we don't get mice born with tumors, or mice born with very abnormal growth defects? So that was the first question. Okay, and maybe if there's somebody who's answered that in the chat box, we can ask if anybody amongst our 5 students would like to answer. Anybody would like to answer it? Prasanjit, I don't think even remember the questions, because he was not one of the students and he didn't take it down. Okay, and he didn't go back to the recording. Chitra didn't come to this one, right? But you can attempt it, Chitra.
Yes, ma'am. The loss of this RB and P53 genes, they will be compensated for by some other genes.
Yes, that is so correct. Okay, and what have they said in the chat box? Have they said anything in the chat box?
Yes, my chat box, RB and P53, there's a compensation. Other DNA repair pathways take over their roles. They are more important for preventing tumors, but not normal embryonic development. It's true that they control the progression. They will definitely be prone for development of tumors, but they will not affect normal growth.
Growth and development. Yes, I think that's an extremely sort of good answer, and we will just discuss it a little bit in detail a little later. Okay, now we come to the next question, and we'll take them one by one. Okay, the next question I'll just enunciate it, and that is, why are only a few organs the targets of cancer in the inherited syndromes? Okay, and I'm sure that people have answered this, and I'll just go to the next one, and then one by one we will discuss the answers to these questions. Why is P16 a surrogate marker for HPV in some systems? Okay, last time we talked about HPV, which is a very important tumor. Is a very important agent, yes, for tumors, particularly cervical cancer. And finally, the last question was, what is the pathogenesis of squamous carcinoma? All right, so now let's go to this first thing. Okay, And this is the answer to that question. And the answer was very well given by Chitra and all those who answered in the chat box. And that is, nature has a huge amount of redundancy in the system. And so we have to think very seriously about, we talk about tumor oncogenes and we talk about tumor suppressor genes. And then when these genes are knocked out, you don't get tumors, or if you get excessive numbers of sometimes of oncogenes, you may not get tumors. And we have to ask ourselves why. Okay, and this is true what you said, but it's strange over here. Okay, if you knock out the RB gene in a mouse, you know what happens? It will not develop at all. And you see the second bullet point over here. The mice will only develop normally if this gene is replaced in the trophoblastic cells. And you know that the trophoblast, well, then maybe I can ask Harshad. Harshad, what is the trophoblast? Are you there, Harshad? What is trophoblast?
During embryogenesis, the formation of the cells.
Embryogenesis, the formation of cells. Is that what is a trophoblast? Yeah, is that what a trophoblast is? Is it? All right, anybody else over there? Apart from Chitra, because she's a third year student and she's answered. Sony, what is trophoblast? Sony, are you there? Sony seems to
Tony Kumari, unmute yourself, please.
She just disappears. Okay, she's not there. Okay, Harshavardhini, what is trophoblast?
Ma'am, trophoblast is an outer layer of an early embryo, the blastocyst. So it forms an outer layer. It has two epitheliums. One is syncytial trophoblast and the other is cytotrophoblast.
You think that the trophoblast is the outer layer of a blastocyst? Really?
Yes.
Oh, okay. What? All right, let's see. Sony's not there. We have our Prasanjit. Prasanjit, what is trophoblast?
Cells which form the placenta.
Yeah, Prasanjit is absolutely right. Trophoblast is that part of the embryo. Yes, it's not actually a part of the embryo. The zygote gives rise to when the zygote divides, it gives rise to two different parts. One is that will go to form the embryo, and the other is the extra embryonic part. And in the extra embryonic part, and it is part of the zygote. It is not part of the maternal. It's not part of the mother at all. Okay, and so trophoblast is the extra embryonic part of what happens when the zygote starts to divide. Okay, there is another extra embryonic part, and that is the yolk sac. So you have two extra embryonic portions. One is the trophoblast, and the other is the yolk sac. Now the trophoblast goes on to be part of the placenta, like what Prasanjit said. But Prasanjit, you're right. But you tell me, in this placenta, what parts do you have? You have the trophoblast, and what else do you have? Yeah, what else do you have? So is the entire placenta part of the fetus? Is that so? Or is there something else that contributes to the placenta? What do you think, Prasanjit?
Part.
Huh? What?
Part.
What? Sorry, Prasanjit?
Part of embryo.
It's a part of the embryo. I'm asking you, what is trophoblast? You said it is a part of the placenta. Is there any other part to the placenta except for trophoblast? Yeah, there's lots of people answering in the chat box. Yeah, there is a maternal part of the placenta as well, right? So trophoblast is part. It belongs to your zygote. It belongs to the fetus that is developing. Yes, but there is a maternal part to the placenta as well. So Prasanjit, you were right, but you didn't understand what the placenta actually is. Yes, the placenta has a maternal part and it has a fetal part, and the trophoblast belongs to the fetal part. Okay, to come back to these mice, there will be no development of an embryo at all unless in these RB null mice, unless you replace, and it's possible to replace, they replace the RB gene, okay, the double dose of the RB gene in the trophoblast. If they do it, then a placenta will form and there will be absolutely no problem with embryogenesis. You know that unless in mammals, and I hope you know that a mouse is a mammal, okay? You know that mice suckle at their mother's teats. That's why a mammal is called a mammal, because we have mammy. Mammy means breasts, okay? So a mouse is a mammal and the mouse has a placenta. And unless there is a placenta, no embryo can develop. So if you allow that placenta to develop, then the RB gene is very crucial in the development of the placenta. So if you allow that placenta to develop, then the embryo has no problem in developing, and it develops into a completely normal embryo. Okay, it's all about redundancy in the system. Now, with respect to P53, there is no problem at all. Embryogenesis goes on very well, because it has no effect either on the trophoblast or on the embryo, on embryogenesis. But if those mice live long enough, yeah, they may develop tumors. Okay, so as far as normal development is concerned, the P53 gene is redundant. But once there is an adult that develops, then the P53 gene becomes relevant for repair and regeneration and whatever else, apoptosis in the adult mouse. Okay, very good. Now the next question: why are only a few organs the targets of cancer in the inherited lineage? What are the answers to that? Yes, Chitra, I'm asking you because you are ahead of most of the others because you have been longer as a postgraduate student. Yes, tell me.
It's only in some organs.
Yes, why? Is it only that gene is knocked out or is abnormal only in those organs? You know that if you have a retinoblastoma, a familial retinoblastoma, yeah, Chitra, every single cell in that body has lost, either by deletion or by mutation, the function of one retinoblastoma gene. Yes, because that has been inherited from one parent. Correct, Chitra? What I said, is it correct? Chitra, you disappeared? What happened to Chitra? Oh, you can't hear me. Have I disappeared?
I hope I'm audible.
Can you hear me?
Yeah, I can hear you.
No, you're not audible now.
I'm not audible now?
No, ma'am, she's not audible.
She is not audible. All right.
Am I audible now? Am I audible now?
Chitra, very faintly.
Okay, ma'am. I heard the question. Can you hear me now?
Yeah, just about, yes.
The RB gene,
Yes.
you inherit one from each parent.
It doesn't get one from each parent. It gets two. Yes, it gets, but one is abnormal, no? So it has come from one parent, the abnormal gene. Then the other gene is normal and it's come from the other parent. So every cell in my body, Chitra, has got the abnormal gene, right?
Yes.
So why do I not get a skin cancer or a bone cancer or some other cancer? Why?
Because some organs are more prone to
So what do you mean by more prone? Why?
The-
Because the RB gene is there in every cell and it has an important role to play in the cell cycle. So what do you mean by prone?
those organs, some organs are not able to compensate for the loss that has happened.
They're not able to compensate. Meaning what? Meaning what? How do they compensate?
Some specific insults that are there to the organs.
All right, fair enough. I think I get your drift. Is there anybody else over there who wants to say what have they said in the chat box?
In the chat box, they said because of specific functions of mutated genes in one. So there's a difference in the gene activity. Means one gene can be highly active in one tissue, but not or less active in the other tissue.
Yes, so that's I think
Like
what
you explained in RBG.
Yes, and I have given the 2 bullet points over here. And if you look at it, you say that the vulnerability of cells and tissues, are according to where the cooperation of other cellular and tissue systems are required at specific time points and specific stages of development. Okay, first of all, we know about redundancy. Now, that answer is very pertinent over here. Okay, once the embryo has developed normally and you have one abnormal gene over there, that gene probably has a very specific role at a specific stage of development in that particular organ. Okay, and if whatever that gene or the loss of that gene can be replaced by other cellular genes or tissues with the help of whatever other tissues are around it, then you won't get the cancer. And this is something that we have to understand. So it is not as if our genes define what is going to happen in our tissues. And we will come to that a little bit later when we're talking about what we call the somatic mutation theory. And you have a lot about the somatic mutation theory in your chapter 7. Okay, so and then cancers initiate the effects of oncogenic events on susceptible cell of origin states. Okay, so you may have every cell in your body has inherited the retinoblastoma gene. Okay, but it seems to be important only in young developing lives, and that too in postnatal life. So in postnatal life, the retinoblastoma gene, although it has a function in every cell in our body, but with respect to oncogenicity, it is very susceptible. It's the developing retina that's very susceptible to the loss of function of the retinoblastoma gene. Okay, but when you take the P53 gene, and you know that there is what we call the Lee-Fraumeni syndrome. Yeah, the Lee-Fraumeni syndrome is a syndrome that occurs when you inherit a P53 mutation in the germline. And according to Knutson's hypothesis, the other gene will also have to be knocked out, deleted, or mutated if you have to get a cancer. This is what happens in, according to Knutson, in whether you have a sporadic cancer in which you get one P53 knocked out during in a sporadic event, and then the other P53 has to be knocked out. But in Lee-Fraumeni, you are born, or that individual is born with one P53 knocked out. And because there is one P53 knocked out, the other P53 will have to be knocked out in postnatal life. All right, but with P53, because P53 is so active in so many tissues, you have a susceptibility to getting carcinomas and sarcomas in many organs and tissues. So if you look at the Lee-Fraumeni syndrome, you will find that these patients get sarcomas in the soft tissue, sarcomas in the bone, they get carcinomas in various organs, females, in the breast, males, in other organs, and that's because the second hit in the P53 deficient or familial P53 Lee-Fraumeni syndrome is the cells, when they get the second hit, are susceptible in many different organs. Okay, so there is a difference between retinoblastoma as well as P53. If you take APC, that is adenomatous polyposis coli, it is the colon and the gastrointestinal tract. The colon is most susceptible. The small bowel is less susceptible. You may even get these polyps, and then they go on to get carcinomas in the stomach. Okay, so it depends on, as Chitra said, and what people were talking. It is some particular organs have a susceptibility to these to oncogenic changes, depending upon which tissues and organs have got the propensity to have these cancers. So you can't say that because you have inherited some kind of quote-unquote oncogenic or tumor suppressive genes that any or every one of your tissues is at risk. They are not. Okay. Now, here you are. Why is P16 a surrogate marker for HPV in some systems? And let's take cervical cancer as a model system. Has anybody said anything in the chat box, or anybody here would like to answer? Chitra, you are aware?
Yes, ma'am. So the HPV, it is a virus that targets 2 proteins, that is the E6 and the E7. Yes. And when the action of the E6 gets affected,
yes.
There is...
yes. What does E6 do?
It helps in the
cell cycle. You can see this over here and you can read it because you're right. The virus produces, and this is not all HPV, it's the oncogenic or what we call the high-risk types of HPV. We get lots of warts and other things which are caused by HPV virus, but they are harmless variants. But there are high-risk variants, and Chitra is right. So here you can see Chitra, so you can you know what HPV6 does, and you can read this off this slide.
P16, it is required for cyclin D1 to properly function, and the cyclin D1 is required for the function of RB gene, and that RB, the G2M transition.
Yes, okay. So it's in simple words, HPV6 produces inhibits P53 action. Okay, that's what E6 does. E7, on the other hand, inhibits RB function in the sense that RB, when it is bound to E2F. E2F is a protein that prevents transcription of the gene. Okay, so whatever that transcription and E2F prevents this cell from going into S phase. So it stops replication. Okay, so it's simple. So HPV E7 prevents this from happening. Okay, and here you see on the left hand side, when you have the retinoblastoma gene, and here you have the retinoblastoma gene, this little triangular outside coat, and you have the protein E2F, that retinoblastoma protein, because the gene is in the DNA. So this is the protein which has bound itself to E2F in a hypophosphorylated state. Okay, what does HPV E7 do? It releases this RBE2F complex, and then you get the transcription and you get promotion of the cell going into S phase, and that's what happens. Cells start to turn over. We require the retinoblastoma gene to be bound so as to E2F, complexed with E2F, so that it does not promote the cell from going in from G1 into S. Okay, now this E7, because it prevents it from happening, you see over here that there is a build-up. Okay, and it does this, as she was very rightly saying, by promoting this increase in the CDK4 cycling D. Okay, and when you do that, normally when you get this increase of CDK4, the P16 protein, the gene that produces the protein P16, immediately gets into the system to try and suppress this CDK4, because CDK4 is a promoter of growth and pushes the cell into the S phase. And if it is not happening, and you will get a buildup of P16, because P16, the cell is producing a lot of P16 to try and stop this increased CDK4 and cyclin D from promoting the cell into the S phase. So because the feedback mechanism is cut out, you will have a huge amount of P16 building up inside the cell. It's trying very hard to stop cyclin D1 from promoting growth in the cell, to pushing the cell into S phase. It's trying its best, but it can't do it. And the cell is producing more and more P16 because it wants that S phase to stop. But because you have E7, which has come into the system, it makes no sense, because it doesn't. This CDK4 is hypophosphorylating the retinoblastoma gene, and this hyperphosphorylating it, and this retinoblastoma gene is then released from its complex with E2F. Okay, and now that's why this is a very interesting buildup of P16, because it helps us. It helps us in the lab. Sorry, I don't know where that has gone. But anyway, I will show you what happens with P16. I think it was in the last time's talk. When P16 builds up, we can now have an antibody to P16. And in a cervical cancer, whether it's an adenocarcinoma or a squamous carcinoma, in an oropharyngeal cancer, which is usually a squamous carcinoma, if I find huge amounts of P16, I know that there is HPV that has infected those cells. All right. So now we have the next question. What is the pathogenesis of squamous carcinoma? And this, I think, any one of the students who are here, and let me ask, is Sony there or she's not there? Sony, are you there? Can you unmute yourself? I think we can more or less take it that Sony is not going to answer. All right, Harshad, what is the pathogenesis of squamous carcinoma?
It could be because of some pre-malignant lesions like CIN.
Pre-malignant conditions like CIN, all right?
Yeah. Or few major mutations like TP53 or Notch.
What causes CIN?
HPV infection.
Infection, okay. So do you understand, Harshad, that built into this question are really two questions? Can you think what are the two questions that are built into this one question? And that is, what is the pathogenesis of squamous carcinoma? Think, there are two questions in this one question. Go on. You can't think. Harshad, right from the beginning, I have got the feeling that you are here in pathology because you're not really interested in pathology. Am I right, Harshad? What did you want to be, but you are in pathology? What was your first choice, Harshad? Harshad, what was your first choice? You want to be a surgeon?
Pathology.
What was it? Pathology?
Yep.
Really? Wonderful. Then you're not really thinking like a pathologist. Okay, like you want to be a pathologist. All right, Harshad can't think. Let's ask Harshavardhani. Is she there? Or is she also disappeared? Do we have Harshavardhani?
Yes, ma'am, I'm here.
Very good. So what are the two questions that are built into this one question? What is the pathogenesis of squamous carcinoma?
So there are many pathogenesis related with the squamous cell carcinoma, ma'am. It might be due to environmental or genetic factors. One is
okay.
One
question. 1 minute, Harshavardhani. One of the questions that is built into this is what is the etiology of squamous carcinoma, right?
Yes, ma'am.
So one of the questions that is built into this one question is what is the etiology of squamous carcinoma? Very good. What do you think would be the other question?
The other thing may be mutations, ma'am. Either it may be tumor suppressors.
Okay. All right. Anybody can think of what would be the other question that is implicit in this question? Anybody in the chat box is saying anything?
Ma'am, is it risk factors, ma'am?
Risk factors is etiology, right? Yes. So what else? It is where does squamous carcinoma occur? Because when we are talking about the pathogenesis of squamous carcinoma, the first thing that should strike you is that squamous carcinoma doesn't occur only in one place, right? Yeah. There must be different etiologies or different risk factors operating in different areas of the body where squamous carcinoma occurs. So unless I know what or where the squamous carcinoma is, Yes, it's not like saying what is the pathogenesis of undifferentiated nasopharyngeal carcinoma. Yeah, what's the pathogenesis we know? Oh, what is the pathogenesis of undifferentiated nasopharyngeal carcinoma? Yes, Chitra?
HPV.
It's not HPV, it's EBV. Yes,
EBV.
and we know what happens when EBV gets into the system, how it gets in over there, you know, and what it does within the nucleus. So there's only one thing that produces undifferentiated nasopharyngeal carcinoma. Will somebody please stop talking on the? So you mute yourself if you're going to talk to somebody. Okay, I don't know who that was. Whether
Prasanjit.
Yeah, Prasanjit or whoever. All right. So now, Prasanjit, we have two questions over here in this. What is the pathogenesis of squamous carcinoma? Number one, what are the sites at which squamous carcinoma occurs, and what is the etiology at each site? So can you tell me, Prasanjit, what are the sites at which squamous carcinoma occurs?
Please log out.
Prasanjit has logged out.
Oh, he's logged out. All right, very good.
He has
So,
some network
who do
issues.
we have now, Harshad? Can you tell me?
Yes.
Yes, tell
Yeah.
me. Tell me where does squamous carcinoma occur?
Skin, mostly.
Skin, very good.
Cervix,
Cervix, okay.
buccal mucosa, that is.
Okay, buccal mucosa.
Yeah.
That's it. That's it. Okay.
Ma'am,
Harshad?
we may have yeah, ma'am. We have bladder, ma'am. Anorectal SCC, ma'am.
Okay,
we may have
bladder, we have what?
then we may have lung, ma'am. Larynx.
Rectum, yes. Lung, very good.
Larynx.
Yeah, larynx. Anywhere where you have squamous mucosa that occurs either primarily or after metaplasia. That,
Yes, ma'am.
so in the pathogenesis of squamous carcinoma, you have to factor in metaplasia. That's extremely important, right? Somebody saying where there is something. I didn't catch that in the chat box. So look at this. Look at the places where squamous carcinoma occurs. Okay, in the skin, due to ionizing radiation, chronic scarring and inflammation. Yeah, you have burns or you have varicose ulcers that are long-standing. In the cervix, it is HPV. In the nasopharynx, it is EBV, because undifferentiated carcinoma of the nasopharynx is a form of squamous carcinoma, and you do have squamous carcinomas of the nasopharynx which are caused by EBV. Then you have the nose and paranasal sinuses, and you get squamous carcinoma there induced by chemicals or by tobacco. Oropharynx, HPV, and tobacco. Buccal mucosa, gingival mucosa, smoked and smokeless tobacco, and to that maybe a cofactor, that is submucous fibrosis, which is areca. Then you look at tongue, larynx, lung, esophagus, urothelium. All of them are tobacco. In the urothelium, you might have chemicals, but even without them, if you have chronic calculous disease, both in the bladder as well as in the venal pelvis, you will get squamous carcinoma by all. Now look at this, lung and urothelium. Yeah, lung and urothelium do not have squamous mucosa, and yet you get squamous carcinoma. So how do you get squamous carcinoma in the lung and in the urothelium? The others all have squamous mucosa. So what is the answer to that question, Harshad? The lung does not have squamous carcinoma in the bronchus. Urothelium has urothelial epithelium.
Metaplasia.
So the answer is metaplasia. So implicit in this question of what is the pathogenesis of squamous carcinoma is it differs depending upon what the site is, what is the etiology, and how that etiology actually acts in the individual site. It might do it through metaplasia. It may do it through other things, like for instance in the skin, UV radiation. What is it doing? It is causing DNA damage. Okay, tobacco, yes, is also doing DNA damage. HPV is not damaging the DNA. What is it? And we just talked about how HPV induces the cell to undergo repeated growths and S phase transition because of the E6 and E7 virus. So E6 and E7 proteins of the HPV virus. So just be very careful. Somebody or some teacher who may be listening to this talk may ask a question in the exam, what is the pathogenesis of squamous carcinoma? And you better know what it is, okay? Because it is an extremely important a disease to study from the oncogenic point of view, because there are so many different etiologies, so many different ways in which those causative or risk factors can act on the individual cells which will produce the squamous carcinoma. So now that we're talking about squamous carcinoma, I think, and of course this is what Harsha just answered, okay? We come to epidemiology and carcinogenesis, okay? And epidemiology is the study of, it depends on what you're studying as far as epidemiology is concerned. You can study the epidemiology of anything, including thalassemia, okay? So epidemiology is about studying how a disease, how and why a population has a particular disease. What is an important disease in the population? And as far as the epidemiology of cancer is concerned, we need to find out the incidence by site. That's what we do. And you see a little cartoon here, and I think every postgraduate should know what is a population-based cancer registry. Does anybody here know, Harshad, what is a population-based cancer registry? I have only 3 people I can ask, because Prasanjit has disappeared and so has Sony. Yes, come on, Harshad. What is a population-based cancer registry?
Registration of all the cancer cases.
Yes?
Registration of all the cancer patients in the particular area.
Registration of cancer patients. Okay. But what do you mean? Yeah, but how do you do it? How do you do it? Think. Registration of all cancer patients. Okay. What? Yes.
It is a register in which the different types of cancers and the cases, the patient details, those are also included, and it is done.
Okay, all right. So just tell me, Chitra, can you imagine how it is done? We're talking about population based. In a hospital, it's easy, no? Because all your cancer patients are anyway, they are there, they have case papers, they may have window papers, they have all the details. So what do we in a population? So how do you think people go about getting population data on cancer?
By the surveys that are taken and the national
surveys are taken. No, there is a methodology that is used. Okay, every place that treats cancer. Okay, so you may not know it, but if you are in a medical college in a city which has a population-based registry, and there are 38 population-based registries in our country. Once upon a time, there were only 3, a long time ago. it's moved from there all the way to 38. Okay, and they are in different cities across India. You notice there's none in Jammu and Kashmir. For whatever reasons, there's no population-based cancer registry in Jammu and Kashmir. The first population-based cancer registry, I am proud to say, was the Bombay Cancer Registry. And it was under the auspices of the Indian Cancer Society that this cancer registry was built. So what do these registries do? They have field workers. And the field workers go out into all the hospitals and all the major teaching hospitals and major institutions and clinics which treat cancers. Okay, because cancers cannot really be treated in small nursing home. Maybe a biopsy is done in a small nursing home. Maybe sometimes a simple kind of excision or section is done in a small nursing home, but by and large, these are done in big institutions. So thus, all of these field workers from the Bombay Cancer Registry, and it happens in Trivandrum and it happens in every place where you have a cancer registry, these field workers go out and get all the information from these places. They also go to very big pathology centers, because pathology centers are where these cancers are diagnosed. Okay, it's not in small private private pathology labs, and if they are diagnosed in private pathology labs, then that patient will be referred to a major cancer center. And then when they collect all this information, they start to look for duplications. And this is very hard work. Once upon a time, it was done by hand, by scrolling, by looking, etc. Now it is, of course, done by computers, and they try hard to see that there's no duplication of data, and you get a record that is put into the registry of these cancer centers. Of the individual registries. Okay. So now, you want the incidence by site, you want to know the age, and for women, it is almost always that they want to know if the women are menopausal or premenopausal, okay? Because that is important for many of these cancers like breast cancer. Things that this epidemiology actually impacts, once you know that there is a high incidence of a particular kind of cancer in a particular area, then the research goes into why is there this particular cancer is very common in that area. And those of you, I don't know if there's anybody in the chat box who comes from the north, Bihar, UP. Do you know what is a very important cancer that you get there, which we see very little of in the south and certainly very little? I don't know if you know, but it is gallbladder cancer. Very common. And it's very common in the Indo-Gangetic plain. And there is a lot of research to find out why gallbladder cancer is involved in, yes, Manisha says oral and whatever. But yes, there is gallbladder cancer, which you don't see almost anywhere outside of the Indo-Gangetic plain. So you, Bihar, going into West Bengal, you see gallbladder cancer. So the epidemiology then gets into environment and lifestyle, what kind of chemical exposure, radiation exposure, UV, tobacco, alcohol, diet. Virus is very important, and of course, genetic predisposition. All right, I won't go into this, but just to tell you that once upon a time, cervical cancer was the leading cause of cancer in women. In our Bombay Cancer Registry, it is now breast. But when you go to Mizoram and you go to Tripura, you still have very high incidence of cancer of the cervix, and this gives you the number, and that number is how many cases do you get per 100,000 population. Okay, you see that cervix uteri is 10.4 here. Look at the breast, 32.7, and if you go down to breast in Tripura, it is 6.9. Since you come from the south, you might like to know that in Trivandrum, breast has also overtaken cancer of the cervix, and it's a high incidence now, 36.9. And then research goes on as to why this happens. We don't have the time to talk about it now, but I'm telling you that this is a legitimate question that you can be asked either at the orals or even in your theory papers. Okay, so if you're postgraduates over here, you're going for your exams, you better do some reading on Indian cancers, where they occur, what is their propensity for occurring, and the epidemiology of those cancers. All right, now we will come now to neoplasia part 2. Okay, and just for the sake of people like Prasanjeev, etc., who never attended the last time, this was the first slide of my last of neoplasia part 1. And this is what we understood about cancer. Okay, we had a definition of cancer, which is here, and which Prasanjeev gave me when I was just talking to him before we started this class. Sorry. Okay, then there is this other things that we understood about cancer, and that is it is a neoplasm that arises from the clonal expansion of a single neoplastic cell. That cancer is a malignant neoplasm. Sorry, cancer is a malignant neoplasm. That cancer is a multi-gene disorder caused by somatic mutations. I'm sure all of you have heard this. All of you have read this in your textbooks. Cancer is defined by hallmarks, and that there are consistent differences between normal cells and cancer cells. And a cancer cell is definable. That means you can define each cancer cell. Okay, so this is our common understanding of cancer. And then we looked at the definition of a neoplasm, and we found that there were many exceptions to the rule in this definition of a neoplasm. Yeah, and we discussed this at length, that a uterine leiomyoma, according to this definition, is not a neoplasm because it is not independent of physiological regulatory stimuli. Okay, what about an extra thumb? It is abnormal, its growth should not have been there, and it exceeds, it is uncoordinated with that of normal tissue, and it certainly persists. Okay, but why is an extra thumb then not a neoplasm? And we answered that question by saying that it is not just uncoordinated growth, but it is in the definition of a neoplasm, there must be this word differentiation. Okay, and so we said, because a thumb looks like a thumb, it is organized like a thumb. And all neoplasms are disorganized. Okay, so in a neoplasm, yes, you must have proliferation, but you must have disordered tissue organization. Yes, you must. Most neoplasms are clonal, but not all need to be, and we will discuss that. And then we ask this question: can an amoeba get a cancer? And the answer is no. Why? Because it is a single cell. Absolutely right. So there is no tissue for it. And then we said that neoplasia is a process that occurs in metazoa. Okay, so I'm not going to go into all of the things that we did the last time. If you want to go and see the recording, you can. But let's now come to this bullet point on our understanding of cancer. We believe that cancer is a multi-gene growth disorder caused by somatic mutations, and we talked a lot about somatic mutations. You know, we talked about oncogenes, and you have EGFR, and all those that we talked about the last time, and how in lung cancer you have EGFR mutations, and so on, which arise from the clonal expansion, which you have one cell that became mutated or neoplastic, and then all its progeny formed the cancer. So now let us examine this particular statement. Okay? And this statement is seen in what you'll see that in your textbooks. There is this theory. It's called the somatic mutation theory, which I just enunciated. Okay, it is the prevailing paradigm. The word paradigm. Chitra, would you like to define a paradigm? What do we mean? Because you see it everywhere. Yes.
The assumption, the working theory.
Yes, it is a theory that explains what we are seeing. So it is the accepted theory that that is general in a particular scientific community. Okay, so that's what paradigm means. And the prevailing paradigm today in cancer is that there are heritable changes that occur in the genome of one cell, which gives rise to a clone of descendants, that constitutes the cancer and display all its biological hallmarks. Okay, and we talked about the 10 biological hallmarks, which were described by Hanahan and Weinberg. Okay. Implicit in this definition and in this theory is that mutations accumulate in cancer cells over time, because they also say that one of these hallmarks said that there is genetic instability in a cancer. Yes, so mutations accumulate in cancer cells over time and lead to tumor progression. And therefore, according to this SMT, cancer is a multi-gene disorder because multiple genes are abnormal. That begins as a clonal alteration in a single cell. That is, in essence, the paradigm that most people believe, and certainly most cancer research and therapeutics is based on this somatic mutation theory. Find an abnormal gene and target it. Find more than one abnormal gene and target it. That, whether you can use targeted therapy or you just target the DNA and kill the cell. That is what the somatic mutation theory is all about. And I don't need to show you this. This is a very iconic diagram that is in your Robbins. You have a normal cell. According to that, carcinogen or otherwise induced mutation, you get initiated, means this is the cell in which that mutation has taken effect. And that precursor has got stem cell-like properties because it can, you heard all that in the hallmarks. There is immortality, telomerase can be regenerated in these cells, etc. Then you have additional mutations, and then you have a founding cancer cell. This is also a cancer cell, but with the additional mutations, this cancer cell goes on to form the tumor in which additional mutations occur. And finally, although we talk about clonality, yes, these are all the progeny of this one cell according to the somatic mutation theory, but you get, with additional mutations and with these cells being very prone to get mutations, there is instability, genetic instability in tumors. You get a genetically heterogeneous cancer. Okay, it's all there in your Robbins, and we will now try to understand this. Okay, so somatic mutation theory, normal, and it just gets more and more and more abnormal until you get a cancer that is growing. It forms a growth, and in that growth you have a genetically heterogeneous cancer. And I want you to understand. Look at this arrow. It's not an arrow, but it says that the cells get bigger. I mean, the cancer gets bigger and bigger and bigger. Okay, do you see anything happening in the other direction? And the answer is no. Okay. And this somatic mutation theory and all the hallmarks which are shown here, we use this in therapeutics. Okay, we use cyclin-dependent kinase inhibitors, we use telomerase inhibitors, we use selective anti-inflammatory drugs because one of the hallmarks is tumor-promoting inflammation. You have inhibitors of invasion and metastasis. We actually have drugs against it. You have MDM2 inhibitors against this genomic instability, anti-angiogenic factors, pro-apoptotic factors, aerobic glycolysis inhibitors, EGFR inhibitors, you know that, and immune stimulation. You're using checkpoint inhibitors. And so these are the hallmarks. This is a somatic mutation theory. We have been very successful, and therefore we reinforce the fact that this somatic mutation theory is the right paradigm. Okay, now let's see if that is true. Okay, but there are nagging questions, and I think all of us should always look at the other side of the coin. Otherwise, I think we will sometimes be led astray. And the first thing is, what is the origin of therapeutic resistance to drugs targeted at driver mutations? You have mutation like EGFR, for instance. Okay, EGFR, we're using targeted therapy, and yet therapeutic resistance occurs. So how does this therapeutic resistance occur? The other thing we just discussed, organ specificity in inherited cancer syndromes. Why do we get organ specificity in inherited cancer syndromes? Why do we get organ specificity in any cancer for that matter? Okay, the SMT says that it should be, you've got this abnormality in all cells, so in all cells you should get this particular, you should get cancers, but it doesn't happen. Okay, cancers, it's very interesting. Cancers can be normalized, and we all know about them, and we will take some examples. Cancers can be induced by transplanting normal cells into abnormal locations, and that's very odd because these normal cells do not have mutations. Okay, now you push them into an abnormal location, and they start to form tumors. Cancers respond therapeutically to physiologic stimuli, and we all know that about breast cancer. Okay, and you know that estrogen drives certain cancers, and you can cut that estrogen drive down by giving anti-estrogens, and the cancer shrinks. Normal individuals, now these are normal individuals who harbor cells with the genetic markers for cancer, and yet they do not develop cancers. And I will just tell you that there is a certain number of people in any population, okay, that have the genes for a whole lot of different cancers, and yet they don't have cancers. And a very common one is follicular lymphoma. You have the 14-18 translocation in follicular lymphoma, and there are many individuals, up to 10% in the West, that have this translocation in their normal lymphocytes. They are the lymphocytes, and they don't produce follicular lymphoma. They never had a follicular lymphoma. So why? Okay, they're absolutely normal. Then of course there is cancer dormancy. How does a cancer which is there become dormant? Maybe the cancer has been treated, it becomes dormant, and several years later, suddenly you get a recrudescence of that cancer. So what keeps that cancer dormant? According to the SMT, you cannot make a cancer dormant. It should be proliferating and proliferating because it has the abnormal gene. So something I'm sure is sort of coming to your minds. It certainly should, and it cannot be just with the cancer.
हम इसको पेट पे रखा मैम मैंने भी देखा ना उस जगह से उधर आ गया और ये सर को मत नहीं हुई थी आप उसको वापस हटा भी नहीं सकते मेरे दिमाग में उसका नहीं दिखा था मैं सब कर रहा है हाँ ठीक है आपके दिमाग में इतना नहीं आएगा वही मेरा दिमाग में एक सेकंड के लिए भी नहीं आया अंदर की आप लॉक कर ये हम जाके खाना खाना ये आप खाना खा मेरे दिमाग में ना एक बार भी नहीं आया मेरे से इतना ही आया कि ये पानी में ना गिर जाए
That is afoot. Okay, and then you have to answer this: why cancers occur predominantly in late adulthood, although most mutations occur during ontogeny and early life when cells are actively dividing, and mutations should be very common. Okay, so these are all the nagging questions that occur with this particular paradigm. Okay, so now let us take therapeutic resistance in cancer. Okay, how is this resistance acquired? Now you know we get therapeutic resistance in when we treat bacteria with antibiotics. Okay, you know that. You suddenly get resistance that comes into the system. Everything is is dying off. The patient is responding well to antibiotics, and suddenly you get that particular bacterium becomes resistant to the antibiotic. So how is this resistance acquired? And we know that as far as the bacteria are concerned. Okay? There is a selection of a subpopulation of resistant clones, or if it's bacteria, then bacteria that populate and come to dominate the environment, or that particular, say you're dealing with an abscess or with pneumonia or whatever, when the responsive clones are eliminated by therapy. So if you have something that is sensitive to the bacterium, sensitive to that antibiotic, then those clones of bacteria are eliminated, and lurking around in that background were resistant clones. And the moment the sensitive clones are got rid of, then the resistant clones rear their ugly heads. Okay, and these subpopulations may be small enough to make them undetectable in the clinical setting, because even when you culture, all your organisms, you may not find the ones that grow well, maybe the ones, and you do your antibiotic sensitivity, and you'll find those are sensitive. But once those bacteria, those clones, are killed off by whatever therapy you're giving, those are sufficient to drive therapeutic resistance and patient relapse. Or in the case of the microbiology, you will get a resistant bacterial population. So that is therapeutic resistance. So how? So is it true, therefore, that we have just one clone over there in that whole tumor? Or we have to ask, where do these subpopulations arise? Where do they come from? Okay, and how were they hiding? And we have to ask this question: is it possible that cancers are multiclonal in origin? And it's amazing that the more you look for multiclonality in tumors, the more you will find it. Okay, and I will tell you. See, the trouble is, that methods for investigating tumor clonality have an inherent bias against detecting multiclonality, and I'll tell you how that happens. But despite this, multiclonality has been observed when people have really gone to observe it within all tumor stages, and within 53 different types of tumors that have been investigated, including benign tumors, yes, where it's not dividing very, very fast, and therefore you say, there may be clonal heterogeneity because of fast division, and pre-invasive cancer. So they're not even invaded, and people have found this. We don't need to go into this, but this is there, and this is how you can understand it. Okay, say you have two clones in a tumor. That means there are two sets of progenitors. That means cells that became precancerous, or were the initiators, or could initiate a tumor. Okay, so, and I've just given you an example here. The gray ones is point mutation in exon 21 of the EGFR gene, which is a very common mutation in the EGFR gene. Okay, and then you have another clone, the black one, which is the T790M resistance mutation in exon 20 of the EGFR gene. So it's the same EGFR gene. Okay, one cell, or one clone of cells, has the point mutation at exon 21, and the other clone of cells has got a point mutation in exon 20. They're not in the same cells. They're two different cells. One has got a point mutation in one, and the other in another. Now understand that the point mutation in exon 21 is the one that is sensitive to your anti-EGFR therapy, which is tyrosine kinase inhibitor. Okay? But lurking there in the background are, and of course this exon point mutation in exon 21 is also responsible for driving the growth of these cells, whereas this mutation in exon 20 is doing nothing. It's just those cells are just sitting there, okay, in that particular tumor. You see that as the tumor grows, they're not even increasing terribly in amount, because they are not the mutations that are driving this cancer. This cancer, in the environment in which it is growing, is literally facilitating the growth of these cells which have exon 21. The other ones are just sitting there quietly. They can't do anything very much, because the others have just have outgrown these cells. Okay, now I'll tell you what happens if you get targeted therapy in this tumor against exon 21 EGFR gene, and you find that all the gray cells will disappear. Yes, and what will be left? These other cells, which were not allowed to really grow, they were outpaced by the gray cells. Now they will have a chance to grow, just like your antibiotic resistant bacteria, and then they will rear their ugly heads. Okay, so we have to understand, why is it therefore that we are not able to determine that something is biclonal or triclonal? Why have we for years thought that a tumor is monoclonal? Okay, and the many ways, the most common way in which this has been done is by using this X chromosome inactivation. And I don't know if you know about X chromosome inactivation. We've all studied it. There is a bar body, and the bar body is seen in all female cells. All female cells have a bar body because we have two X chromosomes. In other words, we have double the number of genes, whereas men have a very small Y chromosome, and this has to be compensated for, and therefore we women sacrifice one X chromosome, and that X chromosome is inactivated, okay, during embryonic life. But what happens is that this is a random inactivation. Okay, when X inactivation starts, there are cells that inactivate one X, that means the X might have come from the father, and the other cells randomly inactivate the other X, which is the X that has come from the mother. Okay, and that randomly inactivated X is seen as a very dark dot. It is of course it is chromosomal material, but it is a dark dot that is present in every female cell. And you know that in the old days they used to do scrapes of the oral cavity and look for that bar body to say this is a male or that is a female. And if you had more than one bar body, that means that you were double X or triple X. So one bar body, if you are double X, if you are triple X, you will have two bar bodies, and that of course is an abnormal chromosome. So this is, and Mary Lyon was the one who actually put the bar body on a scientific basis. Okay, so you can have, if you have a zygote and you have random inactivation, one X, this purple one will be inactivated in one cell, and the other one will in the daughter cell, and you'll get the blue one will be inactivated there. And each of these throughout their life will keep the same inactivated X throughout postnatal, even during fetal, postnatal, and adult life. So if during embryological, after the zygote splits, okay, and doesn't happen exactly immediately as the zygote multiplies or divides. You will find it at the 4-cell stage, or maybe even at the 8-cell stage. One or the other X will be inactivated, and the progeny of all those cells will have the same X inactivated for the rest. Now you can understand, no, how you can use this to find out whether a tumor is monoclonal or it is biclonal. And you can only do this in female tumors. Yeah, in tumors, not female tumors, but tumors in females. Why? Because you can't do it in males. There is no inactivation of the X. So you look at the tumor cells, and then by using genes that occur on the X chromosome, and this is one gene that you get, Humara. There are other genes as well. Yeah. You look for those genes, and then you will know whether the gene is inactivated on one X or it is inactivated on the other X, because there is the Humara gene that comes from the mother is somewhat slightly different, or from the father is somewhat different. They're not exactly the same. So you can find out whether this is one X has been inactivated or the other X has been. You can use also the gene for G6PD, okay, because G6PD is also one of the genes that is inactivated, and in somebody who is a woman who's carrying one G6PD deficient gene and one G6PD gene which is not abnormal, you can use the tumor in that particular individual. So this is all, of course, when you're doing research, right? So you have to get tumor samples from these kind of individuals, and then you work on it. It's not something that you do in a lab to make a diagnosis of tumor or anything of the sort. So they found out that they were underestimating biclonality in tumors. And I'll just quickly tell you how this is done. All right, now see this here, that there is a background, and this is a patch. And what do we mean by a patch? It is a group of cells that have got the same X inactivated. And you see that here? This X, okay? It's the small, this is the inactivated X. It is gray in color. This is black. It is perfectly functional. All right. So all of the cells in this patch, all of them have the gray X that has been inactivated. See that? But you see that there are two different clones of cells here. One clone has got this mutation. The other clone has got this mutation. Okay. Same, this mutation. Okay. So there are two clones. But when I sample this tumor, okay, to find out by X inactivation whether all the cells in this tumor or the majority of cells in this tumor have got which X inactivated, because then I will know whether this whole tumor is one clone or not. I assume that it is one clone because the same X is inactivated. But hiding there is the fact that this tumor in this large patch, which are all the cells have got the same X inactivated, there are two clones of cells, and two mutations. This group has got, just notice that they have the same X that has been inactivated. So if I test this tumor, tumor A will be judged to be monoclonal. But now look at this. Here is the patch size in this tumor patch, or the cells that have become tumorous, have grown from two small patches. And if they are two small patches, you see that over here, this is like this. And here, this is the gray X that is not being inactivated and the black X that has. Now, if I test this tumor, I will find that there are two different genes of the X chromosome that are appearing in this tumor. Here, the same gene will be appearing. That means if it's a G6PD gene that I'm testing, all of these will have the G6PD gene that is either deficient or not deficient, as the case may be. Here, I will find, this one has the G6PD. There are tumor cells that have the G6PD gene deficient, and here the G6PD gene is not deficient. And therefore, I'll say, this tumor is biclonal. It may look the same, but in that tumor, you have got different mutations, and this tumor is biclonal. Now that we are doing whole genome sequencing, now that we are doing exome sequencing, and we're not relying on things like, looking at X inactivation, we are finding more and more and more on tumors are not monoclonal. They are actually, many of them are biclonal, and some of them are multiclonal. Okay. And now you've seen the papers that are going up now. And here is one paper. You need not read it, but I'm just giving you the highlights. Our results show that malignant mesotheliomas originate as polyclonal tumors. And we suggest that the carcinogenic field effect of mineral fibers, this is because it's mesothelioma, leads to several pre-malignant clones that give rise to these polyclonal malignancies. Okay, so when you have a big tumor, it may be that there are multiple clones in that tumor. Similarly, with endometrioid carcinoma of the endometrium, they have found the same thing. Okay, and in colon cancers, they say now you have to consider that maybe colorectal cancers are also multi-clone. And the more that we think about it, perhaps we have to go away from this simple somatic mutation theory that there is one tumor cell that gets, abnormal, that gets mutated, or its genome, there's a translocation or some other event that occurs in one tumor cell, and the progeny of that tumor cell then form the tumor. Okay, so I think that we have to get away from the idea that growth must develop from a single progenitor cell in order to be considered neoplastic. And we do this all the time. We ask for clonality studies, and we believe that if something is clonal, then it has to be neoplastic. Now, I think that we have to also understand that every clonal proliferation is not by definition neoplastic. And I told you last time, and I asked everybody in the audience, are you a clone? And we had a little discussion about what is a clone. And a clone is nothing but cells in which the genetic makeup of the cell, the genome, is identical in every cell of that particular body. Okay, body, or tumor, or whatever you're talking about. And here you are. Each of us is a clone. I'm a clone. Every one of you is a clone. Clone of the zygote. We are all clones. Yeah, each cell in our body may not look alike, and that's the greatness of this of nature. We have something beyond the genome, and that is epigenetics, that makes us all different. Okay, so some of us, let's say, jokingly, are benign. Some of us are malignant, but I doubt very much that any of us, yes, is neoplastic. I hope not. Otherwise, our definition of neoplasia will have to change. Okay, so multiclonal, clonally heterogeneous, does it matter? I think not. Okay, so now we have established that this clonal expansion of a single neoplastic cell must be rethought. It may not be so. And if that is the case, we have to ask, are there consistent differences between normal cells and cancer cells? And is a cancer cell actually definable at all? Okay, so let's take this. Cancer cells, I told you, can be normalized. So once a cancer, not always a cancer. Here you have a neuroblastoma. Okay, you can have extreme differentiation that occurs in many malignant tumors. And a neuroblastoma, you and I know, becomes a fully, a fully differentiated ganglionoma. Talk about non-seminal germ cell tumors, that is, they used to be called teratocarcinomas, differentiate into mature teratomas. And they were malignant. Okay, so this once a cancer, always cancer is not really, but then you can say, oh, but they are benign tumors. So from cancer, they became a benign tumor. But let's go a little further. Okay, if you experimentally introduce teratocarcinoma cells, obviously this has to be in mouse, into blastocysts. Okay, so this is a blastocyst, that is a mouse blastocyst, and then you put these teratocarcinoma cells into the blastocyst, and then introduce it into the womb of a mouse. You don't get a teratocarcinoma, you get normal embryos. Okay, so it's not even that they become benign tumors; they become normal embryos. Then the transfer of malignant nuclei, because now you can take out nuclei. Okay, you don't have to take whole cells, and these are malignant nuclei that are taken from teratocarcinoma cells. Okay, and they are put into enucleated oocytes. That means oocytes from which the nucleus has been removed, and put the malignant nucleus into that, and you think, now it's going to form a teratocarcinoma or a teratoma. No, it doesn't. It just forms normal embryos. Okay, so this is very strange, isn't it? That you have taken nuclei, you've actually taken cells, and you have been able to tame these. They become normal. And here you are, mouse mammary cancer cells, when introduced into mammary fat pads of mice. That means you take out all the epithelium from the fat pad. Yeah, that's actually mice breasts. And you just have the fat pad. And into that you put cancer cells, mammary cancer cells. You know what happens? They don't form cancers, they form normal tumors. So now this is something that we have to take on board. And we have to ask ourselves, okay, what is it that we were told by this somatic mutation therapy, and what is going on over here? Okay? Alright, that is one thing. Now you know that you can produce a cancer. You can produce a cancer by transplanting normal tissues into inappropriate sites. And they did it. If you take a normal mouse embryo and implant it into normal mouse testes, okay, guess what happens? It produces a teratocarcinoma. Now, these are normal embryos. Okay, you haven't radiated them, you've done nothing. And then you put it into a mouse testis. Mouse testes don't have embryos. They are not meant to make embryos grow. And what happens? You get teratocarcinomas, and this can be kept growing for over 200 generations. All right. Now you have the teratocarcinoma sarcomas. Teratocarcinomas. Then you take these teratocarcinoma cells and you